So our next presentation is going to be from two students that have been working at the Mexican Venom Lab. Marchando and Romnes are going to talk to us about how venom affects proteins in the blood.
Hi, I'm Marchando.
And I'm Romnes.
And we did research that dealt with hemostatic properties in the venom from two Mexican mountain rattlesnakes, Crotalus Aquilus and Crotalus Triseriatus.
So these two rattlesnakes are quite closely related. The Crotalus Aquilus, its common name is the Caracaro dusty rattlesnake, and Crotalus Triseriatus is also known as the Central Plateau dusty rattlesnake. These two rattlesnakes are closely related. They're about the same size. Most adults grow to about 21 inches. The largest specimen that was found was about 27 inches, and these live in the mountainous ranges of Mexico. They usually live about above 5,000 feet, and they are really hard to find because of the territory that they live in.
So before we discuss the research, I'd like to touch on some of the concepts that we dealt with. So snake venom from rattlesnakes is considered hemotoxic to the blood, which means that it's actually poisonous to your blood. So when the surface of your skin is broken, a model for how blood clots on the clotting cascade occurs. It's a sequential activation of a bunch of different enzymes, as you can see here, and it concludes with the activation of thrombin from the enzyme prothrombin, which then will cleave fibrinogen into fibrin, which will then regroup itself and form a clot. And after the surface of the skin is healed, enzymes are released to dissipate it. So thrombin is considered a serine proteinase, which by definition is a protein, an enzyme that breaks peptide bonds in proteins. The snake venoms that we dealt with also contain these serine proteinases, but they're much more effective than thrombin, and they will degrade fibrinogen to such an extent that it's no longer of any use to clotting. This property is what makes the snake venom so toxic to us in the first place, but also what has promise in the medical world for anticoagulant drugs for people who suffer from conditions such as thrombosis and even stroke. From a pharmaceutical standpoint, this can be tackled in many different ways. There's serine proteinases that target in the snake venom that target the calcium-mediated steps that target factor V, but we're focusing on the ones that target the fibrinogen.
So we ran a gel electrophoresis on the crude venom of 3 specimens of Crotalus Aquilus and Crotalus Triseriatus. Over here, a specimen of Crotalus Triseriatus, and over here is the Crotalus Aquilus. Snake venom can contain up to 30 different proteins, including metalloproteinases, serine proteinases, and phospholipases. Basically, what gel electrophoresis is you insert the snake venom into each lane represents one specimen. So you insert it into each singular lane, and then electricity is applied to the gel. This causes the protein to slowly move down each lane and separate larger proteins on the top and smaller ones on the bottom. And basically, what we're looking at are the serine proteinases, and as you can see, they take up most of what the proteins in snake venom are. And what's really interesting is that the two different venoms look a lot alike, but the main difference is the metalloproteinases right here. Crotalus Triseriatus tends to have a lot more metalloproteinases, which contain anti-hemostatic properties, but the venom from Crotalus Triseriatus was a better anticoagulant, which is surprising because the venoms are so much alike. And there are drugs that on the market that contain snake venom. There's Anchor, which is a Malaysian inhibitor. There's the Toxibin, which is made from the common lancehead. And then there's Palbin, which is made from the Western diamondback drug.
So we ran a series of assays on these venoms to test for how active the enzymes were, how they broke down the protein fibrinogen, and how effective they were at doing so. So the first one that we ran was called the thrombin-like serine proteinase assay. So this one tests how active the enzymes are. We have glass tubes known as assay tubes, and we'll put a buffer and crude venom into them. And except for one that serves as the control, which we put distilled water in the same amount as the venom. And we'll put these in an ice bath for 3 minutes, and then we'll put them in a warm water bath for 3 minutes at 37 degrees Celsius, which is normal body temperature. Our goal is to simulate these reactions as accurately as we can in the body without actually doing them in vivo. So we put the substrate into these, and we let them incubate for 5 minutes, and we stagger them 30 seconds. So we'll add the substrate to the control, and then next 30 seconds we'll add to the next one. And after the reaction has reached 5 minutes, we'll put in a termination solution and put them back into the ice water bath, and that terminates the reaction. And each tube will turn, the solution will turn opaque yellow color at the bottom, and the more opaque it turns, the more active the enzyme is. And we can measure this in a really interesting way. We use a spectrophotometer, which is, it measures the amount of light that the solution absorbs, and we can calculate the specific activity of the enzyme from this. And in our results here, Crotalus had reached about 2,000 was the highest for specific activity, which is not terribly high. But we were really surprised where Crotalus topped out at about 3,000 for specific activity, which was 90%.
So the next assay we did was the vibration assay. And the vibration molecule is there are alpha particles, which is the blue. There's beta particles, which is the red. And then there's gamma particles, which are the green particles. Alpha particles are the large ones, and gamma particles are the small. Now, basically what we did was we put vibration and buffer into a heat box so that it warms to the temperature of blood, which is about 37 degrees Celsius. And the buffer is about the same pH as your own blood because we want to simulate the reaction as quickly as we can to what happens in the blood. And so after we reached about 3 minutes for the vibration and the buffer to warm, we took a control and we took a certain amount of the mixture and put it into 10 minutes for time 0. And then after 1 minute, 5 minutes, 10 minutes, and 30 minutes, we added a certain amount of the mixture because we added Venom and we had corresponding labeled at these times. And as the time was reached after we added the Venom, we took the mixture and we put it into the tube to terminate those solutions. After we had these samples, we ran them through a gel electrophoresis. And the control shows us what vibration looks like without any venom added to it. So right here, there's vibration without any venom. And here's alpha particles, beta particles, and here's gamma particles. And basically, after 1 minute, most of the alpha particles are degraded. And then over the course of the reaction, the beta particles slowly degrade, and then the gamma particles don't seem to degrade at all. The difference between Crotalus, Aquilus, and Crotalus Triseriatus is that the alpha particles degrade much faster over the first minute, and so you can tell from this that Crotalus Triseriatus would be the better anticoagulant.
So the last true test we ran was the fibrinogen coagulopathy assay of Venoms, and this is really the true test of how good of an anticoagulant the enzymes are in Venom. So for this one, we used a machine known as a fibrometer. It measures the thickness of a solution and will stop when the solution is formed to clot. So we have human Romnes, fibrinogen, and buffer, and we put them in the wells of this machine, which is naturally programmed to heat up to 37 degrees Celsius natural body temperature. We ran 3 controls and 3 trials. For the control, this just simulates how blood was naturally clot without any disturbance. So we put buffer, thrombin, and fibrinogen together and started the timer on the fibrometer and timed how long the solution takes to form a clot in seconds. And this was around 15 to 16 seconds on average. And for the trials, we put buffer and Venom together, and then we put fibrinogen with this mixture and let that incubate for 1 minute. Theoretically, the enzymes in the Venom should break down the fibrinogen to such an extent that the time it takes for the blood to clot, the solution to clot, when the thrombin is added after the minute, will be much greater, or hopefully not clot at all. And for Crotalus, it was 30 seconds on average. We ran a few initial trials with Crotalus, Triseriatus, and the results were very promising. They were significantly higher than 30 seconds.
So the next thing we did was we ran a high the venom is run through a column, and the column separates each protein out by size and absorbance. So on this axis we have time, and on this one we have light absorbance. And as you can see, the higher peaks are larger proteins, and these are smaller proteins. And so there were 7 peaks in total. And we took this and we ran it through a gel electrophoresis, and this confirmed that our peaks were the larger ones were farther this way, and the smaller ones are over here, because as you can see, there's larger proteins here, and then there's smaller ones over here. And so the peak that we were dealing with is peak 2, and this is supposed to contain all of the anticoagulant properties. And over here we have a ladder, and it basically tells us approximately where the size of the proteins are in that area, so that we can approximately tell the size of the proteins around the area, like around here and over here. And so that helps us determine the size of the proteins and what kind of proteins they are.
So our main conclusions were that the prospects for using Crotalus in an anticoagulant drug were not that favorable, as in the coagulopathy assay it took around 30 seconds to clot on average, whereas the venom of the Western diamondback rattlesnake took about 130 seconds to clot after the fibrinogen was added. However, using Crotalus Triseriatus in an anticoagulant drug is a lot more favorable and probable, as the initial results that we have were significantly better than those for Crotalus, but we have yet to do further testing on it. The main conclusion that we can take away from this is that we really need to preserve the poisonous and venomous organisms in our ecosystem and our world, as already there's many drugs on the market made from these creatures that treat very serious conditions. Our cases that we work with, Crotalus and Crotalus Triseriatus, are very rare and small, and as the other group mentioned, they're going through habitat shrinkage, and they could be a very wealth to the medical world. We'd like to thank Anthony Gambara for being our mentor, helping us run the assays. Anthony Sagula for helping us run the HPLC machine, which we had no idea how to run. Steve Maxey for being in charge of the lab, purchasing necessary materials, milking the snake. I'd like to thank Crystal Meisel for helping us review our papers and giving us good feedback. I personally would like to thank Chesapeake Energy Incorporated and the Exo Energy Foundation for sponsoring my experience here at FSI and funding our research.
And I would like to thank the Oxford Foundation for sponsoring my time here, because without their sponsoring, I wouldn't have had such a great time this summer, and I wouldn't have been able to research the things I did. I would also like to thank the Disney Family Foundation for sponsoring my research, because I love researching the snakes, and I love to learn about the anticoagulant properties and how they can be applied to the medical world, and how they can help people in many ways, and how these properties in the rattlesnake venom can help us in the future, as long as we preserve these animals and use their colors.
Thank you.
So the question is, what made us decide on these?
What? I guess we were doing one day we were just looking at the crude venom from some snakes that we had some venoms that we had in the refrigerator, and Crotalus. We originally started with Aquilus, as both of our research groups did, and we looked at the venom from Crotalus Triseriatus, just as a random venom, and it was actually different than what we expected, so we ran some tests on it, and it proved to be a very good anticoagulant.
Also, like we said at the beginning of this PowerPoint, not much research has been done on these rattlesnakes, so we like to further the research that was done on these rattlesnakes. Carly.
Do you know what went wrong when you ran that last test on the Triseriatus to see how long it
So basically what went wrong was the thrombin was too it was too the thrombin was too diluted. And so for our test, just to see how it would normally clot, it took longer than 15 seconds. So basically our results weren't concurrent, they didn't fall in place correctly, so the thrombin was taking much longer to clot with the venom.
And also we discovered that the venom we were using was also very concentrated, so the results were really, really high, like over 250 seconds. So, but after doing that, we still got some results, even with the that more diluted venom that were remarkably high compared to Crotalus before. Just
Do you know how the venom actually affects
So the question is, how does the venom affect fibrinogen? So basically, the venom degrades the alpha particles in fibrinogen. And once the alpha particles are degraded, fibrinogen doesn't work whatsoever. And fibrinogen is pretty much the last enzyme that is needed in the polypeptide. It doesn't affect thrombin, but the three incarnations that we're working with have thrombin-like activity because they attack fibrinogen, and therefore it stops the clotting because fibrinogen is destroyed.
Did I answer your question?
So I think that you're working with snakes that are all really close to each other. Do you work with other snakes like cobras or rattlesnakes and their venom to treat cancer?
Well,
Instead of just focusing on one snake and others.
so specialists can use other different groups of snakes like cobras or coral snakes to do the same things that we're doing with here. So the main thing is that the snakes we're working with are hemotoxic, whereas cobras and coral snakes, those are neurotoxic. So, and the main snakes that are hemotoxic are vipers, which are rattlesnakes. So there are some more. There's been the drugs that we talked about. Ancord was made with a Malaysian pit viper, so they all are sort of closely related to start with.